What if I told you that the cure to some of the most stubborn, untreatable cancers on the planet isn't, a $1 trillion designer drug?
Right, which is what everyone expects.
Exactly. What if it's just one of the most basic elemental metals on Earth? I mean, I'm talking about copper.
It sounds a bit like science fiction, honestly, but it is very real, very cutting-edge biology.
Welcome to this custom-tailored deep dive designed specifically for you. So today, we have a clear, uncompromising mission.
We really do.
We're cracking open this groundbreaking 2026 article from the prestigious journal Cell, and we're going to completely unpack every single gear, lever, and mechanism of a newly discovered reciprocal biological circuit.
Yeah, we're looking at a completely new form of cell death, right alongside the element copper and, of course, your immune system.
It is a phenomenal triad, and like what these researchers have uncovered, it completely bridges the gap between cellular metabolism and systemic immunity.
It really does. It's a paradigm shift.
Exactly. So imagine your immune system and cancer therapies working as a tag team, passing a biological baton back and forth to completely dismantle treatment-resistant tumors.
And we really want you to visualize that today.
Right. Whether you're prepping for a heavy biology meeting or you're just insanely curious about where cancer research is heading, get ready. We are not skipping the hard science today.
No, we are diving right into the deep end.
We're going to explore every conceivable detail of this process until it absolutely crystallizes in your mind.
So the mechanism at the heart of this paper is something called cuproptosis.
Cuproptosis.
Yeah, it's a newly identified copper-dependent form of regulated cell death.
Right, and just to ground us, I mean, this isn't apoptosis, right? Because apoptosis is the standard programmed cell death we always hear about.
Correct. So when a healthy cell undergoes apoptosis, it, neatly packages itself up into little vesicles.
It's taking out the trash.
Exactly. It gets cleared away by the body without causing any inflammation. It's very quiet.
But this is different.
Cuproptosis is entirely different. It relies on a very specific, rare cellular mechanism driven by a protein called FDX1.
Right, FDX1 or ferredoxin-1. Let's zoom in on FDX1 for a second.
Sure.
This protein lives in the mitochondria, right?
Mm-hmm.
The energy-producing powerhouses of the cell. And FDX1 is basically a factory manager.
That's a great way to put it.
Its specific job is to facilitate a rare post-translational modification known as protein lipoylation.
Yes. So think of lipoylation as attaching specialized chemical tags, like lipoic acid molecules, onto very specific pieces of mitochondrial machinery.
Right, the factory equipment.
Exactly. And the most important piece of machinery it tags is an enzyme called DLAT.
DLAT.
Yeah, DLAT is a vital cog in the TCA cycle, which, as we know, is the main metabolic engine that produces energy for the cell.
So FDX1 is the factory manager walking the floor, slapping these critical lipation tags onto the DLAT machinery so the factory can keep churning out energy for the tumor.
That is the baseline, yeah.
That's normal operations. But here is where the therapeutic intervention completely derails the factory. The researchers in this study, they introduced a drug called elesclamol.
Which is a copper ionophore.
Copper what?
Ionophore. In simpler terms, elesclamol is a molecular delivery truck.
Ah.
Its only job is to smuggle copper across the cell membrane and shuttle it directly into the tumor cell.
And not just any copper, right? But monovalent copper, or Q plus sire.
Precisely. And this is where the mechanism gets, well, violent.
Violent how?
These Q plus ions act like highly aggressive biological magnets. They are irresistibly drawn to those exact lipoylation tags that the FDX1 manager just placed on the DLAT machinery.
Oh, wow. So they target the tags.
Yeah, and when the monovalent copper binds to these tagged proteins, it forces them to aggressively clump together into a rigid mass. In cellular biology, we call this oligomerization.
Oligomerization. So the copper magnets attached in the tags, and suddenly all the pristine factory machinery gets violently smashed together into a tangled, useless lump of protein.
It does. It's a complete mess. And if that wasn't enough, this sudden influx of copper simultaneously strips and depletes another vital class of proteins.
Right, the iron-sulfur cluster proteins, or FESS.
Exactly. A key casualty here is a protein called SDHB. So you have massive toxic protein clumping happening on one side of the mitochondria.
And the utter loss of essential structural proteins on the other.
Right. The factory catastrophically shuts down. The cell dies. That is cuproptosis in a nutshell.
Okay, hold on. I mean, we figured out how to poison a cancer cell in a petri dish. But, we've cured cancer in a petri dish a million times. Why is this specific copper death so groundbreaking?
Because of what happened when they tested this copper-loaded L-escalmol on living mouse tumor models.
Okay.
Specifically, they used a colon cancer model called MC38, and they tested the drug on 2 groups of mice.
Right, let me guess the groups.
One group was immunodeficient, meaning they had no functioning immune system, and the other group was immunocompetent, with a fully intact immune system.
Let me guess. It only worked in the mice with the immune system.
It worked exponentially better in the immunocompetent mice.
Wow.
Yeah, the drug alone wasn't enough. The immune system was actively required to finish the job.
Okay, let's unpack this. Which means killing the cell with copper isn't just taking out the trash. It's setting off a burglar alarm for the whole immune system.
Yeah.
But wait, how does the immune system actually fight back?
Well, it happens because cuproptosis is an immunogenic cell death, or ICD.
Right, an ICD.
Remember earlier when we said apoptosis is a quiet, neat death? Cuproptosis is a screaming, messy death. As the cell breaks down, it releases massive amounts of DAMPs.
DAMPs. Damage-associated molecular pattern.
Exactly.
So DAMPs are basically a biological flare gun. The tumor cell's internal factory is exploding, and as it goes down, it shoots off these flares into the surrounding tissue to signal that something is terribly wrong.
That's a perfect analogy. And the researchers proved this by measuring 4 specific DAMP flares.
What were they?
They saw extracellular ATP spilling out, a protein called HMGB1 being released, a protein named calreticulin suddenly getting exposed on the outer surface of the dying cell, and the secretion of a powerful immune-signaling cytokine called interferon-beta.
Okay, so the flares are up in the air. Who responds?
The immune system's first responders, which are the dendritic cells, or DCs.
Right.
Dendritic cells are antigen-presenting cells. They constantly patrol the body's tissues, and when they see these DAMP flares, they rush into the tumor microenvironment.
And to actually observe this, the researchers used cytoff analysis, right?
Yeah, they did, yeah.
For anyone not living in a lab, cytophase is essentially like a high-tech biological satellite map. It uses mass cytometry to look at millions of individual cells and their specific protein markers all at the exact same time. It lets you see exactly what the immune cells are doing.
And what the cytosatellite map showed was that these dendritic cells weren't just showing up, they were actively maturing.
Maturing?
Yes. They upregulated specific maturation markers on their surface, specifically CD40 and CD80.
Okay, so what does maturation actually mean here?
Maturation is critical. It means the dendritic cell has consumed the dead pieces of the copper-poisoned tumor, processed them, and is now ready to present these tumor antigens to the heavy hitters of the immune system.
Which are the CD8 plus T cells.
Exactly.
It's like a bounty hunter taking a photograph of a destroyed enemy and bringing it back to the general army to say, this is what we are hunting, go get them.
It's cross-priming, right?
That's exactly cross-priming. And the Cy2F data revealed one more massive shift.
Oh, is that?
Solid tumors are usually packed with tumor-associated macrophages, or TAMs. Normally, these macrophages are corrupted by the tumor into a pro-tumor immunosuppressive state known as M2-like.
Right, they act like bodyguards for the cancer.
They do, but the intense DAMP flares released by cuproptosis actually reprogrammed these macrophages. It flipped them from the corrupted M2-like state into a highly aggressive, pro-inflammatory tumor-fighting state called M1-like.
That is wild. Okay, before we go any deeper, let's do a quick mental bullet point check to lock this in for you. Good idea.
Bullet 1. Cuproptosis uses copper and the FDX1 enzyme to clump proteins together and utterly destroy the cancer cell.
Yes.
Bullet 2. It's a noisy death, an immunogenic cell death that shoots off DAMP flares, rewires macrophages, and wakes up the dendritic cells and CD8 plus T cells.
Perfect summary.
Which leaves us with a massive question for you. If copper kills cancer cells and wakes up the immune system, what happens when the immune system starts talking back to the tumor?
To answer that, we have to follow those activated CD8 plus T cells. Once the dendritic cells cross-prime them, these T cells become activated effectors.
It's activated.
They march into the tumor, and their primary weapon is a cytokine called interferon-gamma, or IFN-gamma.
Now, my instinct tells me IFN-gamma just acts like acid and mucks the tumor. But that's not what the paper says, is it?
Not at all. IFN-gamma isn't a blunt instrument. It's a highly precise signaling tool that specifically sensitizes the tumor cells to more cuproptosis.
This is where it gets crazy. It forms a biological feedback loop. Break down the exact pathway for us. How does a T-cell cytokine make a tumor more sensitive to a copper drug?
Let's trace it step by step. The CD8 plus T cell releases IFN-gamma. This IFN-gamma binds to specific receptors on the outside surface of the surviving tumor cells.
Okay.
This binding event triggers an internal alarm system inside the tumor cell called the JAK-STAT1 pathway.
Okay, the JAK-STAT1 pathway. What happens next?
A protein called STAT1 gets phosphorylated. This means a phosphate group is chemically attached to it, which acts as an on switch.
Right.
This activated STAT1 then travels straight into the cell's nucleus, where the DNA is kept. Once there, it upregulates the expression of another protein called IRF1.
IRF1, which is interferon regulatory factor 1. And IRF1 is a transcription factor, right? It's a protein that can physically bind to DNA and turn specific genes on or off.
Exactly. IRF1 directly binds to the promoter region of the FDX1 gene. By binding there, it forces the tumor cell to transcribe massive amounts of new FDX1 mRNA.
It forces the cell to manufacture more of the FDX1 enzyme.
Yes, exactly.
Let me give you a mnemonic for this, because this pathway is the absolute crux of the paper. Let's call it the signal relay.
I like that.
Imagine the T cell's IFN-gamma is the general on the battlefield shouting an order. STAT1 is the radio operator who receives the transmission. IRF1 is the messenger running the order down to the factory floor.
And FDX1 is the factory manager making those lipoylated tags.
The copper magnets. The general orders the factory to go into absolute overdrive. More FDX1 means the cell is now hypersensitive to the copper drug we are giving it.
That is a phenomenal way to visualize the STAT1, IRF1, FDX1 axis. But this brings us to a crucial biological paradox in the research.
I love a good paradox. Lay it on me.
Remember earlier when we explained that cuproptosis causes the depletion of iron-sulfur, or FeS cluster protein?
Yeah, the SDHB structural supports crumble. The factory falls apart.
Well, FDX1 itself is an FeS cluster protein.
Wait, so you're saying that during cuproptosis, FDX1 is essentially destroying itself?
Yes. FDX1 is absolutely required to start the process of cuproptosis. But as the copper floods in and destroys FeS clusters, FDX1 levels naturally plummet.
Oh, wow.
Theoretically, this should act as a biological break. The tumor cell should quickly run out of FDX1, stop tagging proteins, and become completely resistant to any further copper-induced death.
So the tumor is essentially hitting its own self-destruct button, but the button melts before the countdown finishes. Why doesn't the tumor become resistant?
Because of the T cells. The T cells are relentlessly pumping out IFN-gamma.
Right.
That IFN-gamma hits the STAT1 IRF1 axis, which physically overrides the tumor's natural break. What's fascinating here is it constantly forces the tumor's DNA to transcribe new FDS1.
So it can't run out.
Exactly. The immune system is basically holding a gun to the tumor's head, forcing it to keep manufacturing the very enzyme that is killing it. It locks the cell in a state of terminal vulnerability.
That is beautifully diabolical. The immune system and the copper drug are caught in this perfect self-amplifying loop of destruction. The drug kills some cells, which wakes up the T cells, which release IFN-gamma, which forces the remaining tumor cells to make more FDX1, making them even more sensitive to the drug.
It is a brilliantly elegant reciprocal circuit. It's immunotherapy and metabolic therapy acting as a single unit.
Here's where it gets really interesting, though. I'm going to push back here.
Okay, go for it.
We've cured cancer in mice 1,000 times. If the immune system and the copper drug are caught in this perfect self-amplifying loop of destruction, why don't these tumors just completely melt away immediately?
Yeah.
What is the tumor's secret defense mechanism? Because cancer never goes down without a fight.
That is the crucial question, and it requires us to look at something called adaptive resistance.
All right, before we dive into the tumor's defense, let's grab our bearings for our second 5-minute recap to lock this in for you.
Sounds good.
Bullet 1, activated CD8 plus T cells release IFN-gamma.
Yep.
Bullet 2, IFN-gamma forces the FDX1 factory into overdrive via the STAT1 IRF1 pathway. Bullet 3, this creates a reciprocal feedback loop of immune activation and tumor death. So, question for you listening. Now that we have this perfect positive feedback loop, what biological shield does the tumor raise to stop the T cells in their tracks?
Well, the tumor doesn't just sit there while the T cells bombard it.
Of course not.
The immense cellular stress caused by the cuproptosis, combined with the intense inflammatory signaling from the IFN-gamma, triggers the tumor to upregulate a very specific protein on its surface called PDL1.
PDL1, the classic immune checkpoint. This is the shield.
Exactly. PDL1 acts as molecular camouflage and a biological shield. When it binds to the PD-1 receptor on the surface of those attacking CD8 plus T cells, it sends a powerful inhibitory signal.
It essentially tells the T cell to power down.
It does. The researchers found that this PD-L1 upregulation forces the T cells into a distinct state called progenitor exhaustion, or TPEX.
EPEX.
Yeah, the T cells are physically still in the tumor, but they are functionally exhausted. They stop producing IFN-gamma.
And if they stop producing IFN-gamma, the signal relay stops. The FTX1 feedback loop breaks, and the tumor survives the copper.
Exactly.
So how do the researchers solve this? What is the actual therapeutic algorithm here?
To overcome this adaptive resistance, they designed a highly synergistic three-part clinical algorithm.
Okay, 3 parts.
First, they use a clinically available nutrient supplement called copper gluconate. Second, they use the cuproptosis inducer, alesclamol, to act as the delivery truck for that copper. And third, they added an immunotherapy checkpoint blocker.
Anti-PD-L1 antibodies.
Yes.
Wait, copper gluconate, like the basic dietary supplement you can buy at a pharmacy.
Yes. And that was a very deliberate, smart translational choice by the authors. By using a readily available supplement combined with known drugs, it dramatically shortens the path to human clinical trials.
Okay, but how rigorously did they test this 3-part combo? Because, I mean, if they just tested it on a basic, easy-to-kill melanoma model, I'm not going to be that impressed.
Oh, they did not take the easy route. They graded the evidence against incredibly rigorous, intrinsically cold, and highly resistant mouse tumor models.
And a cold tumor is one that the immune system naturally ignores, right? It's like a fortress with no doors.
Exactly. They tested it on MMTV-PyMT, which is a spontaneous, very aggressive breast cancer model. They used Panos Day, a pancreatic cancer model notorious for being heavily hypoxic, meaning it's starved of oxygen.
And just to clarify for everyone, hypoxia usually makes tumors incredibly hard to kill because lack of oxygen limits mitochondrial function, which is exactly where our drug is trying to work.
Right, which makes this a very tough test. They also tested it on a lung cancer model called 393P, but perhaps most impressively, they tested it on a model called 344SQR.
Why is that one so impressive?
Because this is a lung tumor line that had been continually exposed to immunotherapies in the lab until it developed acquired resistance to standard PD-1 and PD-L1 therapies.
Oh, wow.
It had evolved to become completely immune to our best current drugs.
That is the ultimate boss fight for an oncologist. So what happened when they hit these unkillable, hypoxic, therapy-resistant tumors with the copper-elesglomol anti-PD-L1 combo?
The results were staggering. The triple combination eradicated the tumors and significantly reduced lung metastases across the board.
Eradicated them.
Yes. And when they analyzed the tumor microenvironment, they found it had completely flipped. It went from a cold, immunosuppressive fortress to a hot, violently inflamed state. The key metric they looked at was the ratio of IFN-gamma to IL-10.
Because IL-10 is a cytokine that suppresses the immune system, right? So a massive spike in the IFN-gamma to IL-10 ratio means the immune system is completely dominating the battlefield.
Precisely. Because by administering the anti-PD-L1 antibody, they shattered the tumor shield. The T cells didn't get exhausted.
They just keep going.
They could just keep pumping out IFN-gamma, which kept forcing the tumor to make FTX1, which kept feeding the copper death loop until the tumor was gone. Blocking the adaptive resistance allowed the cuproptosis immunity crosstalk to reach its full lethal potential.
So what does this all mean? Connect this molecular dance to the reality of human cancer treatment for us.
If we connect this to the bigger picture, it means we can force immunotherapy to work on patients who are resistant to it, simply by inducing this very specific immunogenic metabolic crash.
That is incredible. Let's do one final sub-summary before we pull back.
Let's do it.
Bullet 1. Tumors defend against cuproptosis by expressing PD-L1.
Right.
Bullet 2. Combining a copper inducer with anti-PD-L1 shatters this shield, curing highly resistant cancers in mice. So, to you listening, if we can use a basic element like copper to rewire the immune system, what other metabolic vulnerabilities are tumors hiding right in front of us?
That is exactly the kind of paradigm-shifting question this study forces the scientific community to ask. I mean, this paper fundamentally redefines cuproptosis.
How so?
Before this, we thought of cuproptosis as just a tumor-intrinsic metabolic vulnerability, just a fancy way to poison a cell's metabolism. But this proves it is definitively an immunologically integrated cell death pathway.
It's all connected.
It completely changes how we view the intersection of cellular metabolism and systemic immunity. They are not separate systems. They are a single, highly sensitive, interconnected web.
Which leads me to a final provocative thought for you to chew on. We learned today that this entire beautiful pathway relies heavily on the FTX1 enzyme.
Right, the factor manager.
And FDX1's normal day job is supporting mitochondrial respiration and the TCA cycle, which actively consumes oxygen. We also know they tested this in hypoxic low-oxygen models. But think about the reality of a human patient. What happens if a patient's baseline metabolism naturally fluctuates?
Or their diet changes.
Exactly. Could something as simple as a patient's nutritional status or their systemic oxygen levels from daily exercise directly dictate the success of this high-tech, copper-driven immunotherapy?
It's a huge question.
If metabolism and immunity are this tightly linked, we aren't just treating a localized tumor anymore. We are treating the entire metabolic ecosystem of the patient.
It is a fascinating frontier. It implies that every input, oxygen, diet, elemental trace minerals, could tip the scales of immunotherapy. The deeper we go into the cell, the more we realize how deeply connected it is to the whole organism.
Well said. Thank you for sticking through the heavy, uncompromising science on this custom deep dive with us today. We threw a lot of pathways, cell types, and mechanisms at you, but understanding the intricate gear-level machinery of biology is the only way we truly appreciate the magnitude of these breakthroughs. Remember to always look for the hidden connections and keep exploring. Because when you finally see how the microscopic gears turn, the muddy waters of medicine start to look incredibly clear.